Hi, I'm Megan Clark, and this is my enzyme experiment. To start with the introduction, I'll be showing you the papers and then I will be presenting the information. So this is the introduction. So essentially, catalysts are added to substances so that reactions increase, and the enzymes are proteins that act like the catalysts in order for this to actually happen. And essentially what's happening is the enzyme is lowering the activation energy. And this is really important because living organisms need enzymes to have increased reaction times and also to have regulated reactions. And so basically our experiment is we started by adding a buffer, azocasein, and trypsin into centrifuge tubes to see if there was any enzyme activity done by the trypsin breaking down the azocasein. And for us, we did each buffer having a different pH, which contributed to the reaction of trypsin. The more optimal the pH, the more enzyme reaction we could clearly see by the pellet and by measuring the absorbance level. And so the higher the absorbance level, obviously the more activity there is. And the reaction of trypsin is catalyzed and is done by the protein hydrolysis and is cleaved after ARG and PYR. So the primary role of trypsin in living organisms is to degrade proteins in the small intestine. And this is where we get the information of what is most optimal about trypsin. For example, they are in the small intestine for animals, and they are secreted in bacteria cells for bacteria. And the condition for animals is about a pH of 8 with an approximate temperature of 37 Celsius with a highly variable salt concentration, whereas the bacteria has high variables all across in the pH, the salt concentration, and temperature. And based on this information, we had some questions about it. And so we said, what happens to enzyme activity as pH is decreased to a more acidic pH? Or what happens to the enzyme activity if the pH is not at 8, which is optimal for animals in the small intestine? And our hypothesis is if we change the pH to more acidic, the trypsin will not perform at optimal capacity. So now going over materials and methods. So this is materials, this is methods, this is more materials and methods, and then these are some more explanations that I will go through. So firstly, for materials, we used 5 sample trials worth of trypsin, 6 sample trials worth of azocasein. We used 6 different buffers with different pHs from 4 through 9. We observed pellet and supernatant, we used centrifuges, and we used clean spectrophotometer tubes, as well as using that to measure the absorbance. And we had negative controls for each pH, and in this instance we added water instead of the enzyme, and everything else was the same when it comes to salt concentration and temperature. So conditions were kept the same in that area. We used 3 replicates for each pH as well as a 4th sample for the negative control. And we used these replicates to obtain averages and variations, and basically the more information you have, the more that you can infer, and that was the whole point of us doing more samples. So our methods. So we basically added 450 microliters of a buffer, 500 microliters of azocasein, and 50 microliters of trypsin into 5 tubes with different pHs. Salt concentration was constant. And we incubated them for about 20 minutes at 37.4 degrees Celsius. And then we added 500 microliters of 10% TCA to stop the reaction. And we centrifuged each tube for about 2 minutes with the speed of 12,000. After the centrifuging, we observed the pellet size and consistency for each tube, and we wrote all of that down. And then after that, we took the supernatant and we measured the absorbance level at a 440, and we used water to blink the spectrometer. So TCA is added in order to stop the reaction because it is an enzyme inhibitor. This means that it will cease the reaction of the enzyme, which will allow us to fully observe the changes and see the substrate. Since the reaction has been stopped, we have to centrifuge the tube to pellet the undigested substrate in order to properly read the absorbance level at 440. And reading the absorbance level at 440 allowed us to see how well the enzyme broke down the substrate in the reaction. So we're essentially going to go over the results now and all of our data. So in this first table and graph, this is our numbers of absorbance per each sample as well as our negative control. And then we basically take all of this information and put it into a graph. And already you can see that 9 and 8 were pretty high in absorbance level, and the optimal temperature for trypsin is 8. So this could possibly mean that our hypothesis is on the road to being adequate. Next, we are going to look at the averages as well as the standard deviation, and we're going to look at that put into a graph as well. As you can tell, a pH of 7 actually had the biggest standard deviation, and then a pH of 4 had the smallest, and a pH of 9 with the second smallest. And then based on our p-test, our p-value, and our results, we can conclude that the absorbance at 9 is the only one not significantly different, and the rest of them are. So this pretty much means that 9 is optimal. And then this is the conclusion for our data. So essentially, we were saying that during our experiment, we tested enzyme activity with different pH level, and our hypothesis was that trypsin's optimum pH for the small intestine is 8, so we figured it would be optimal in this reaction as well. And our experiment, even though we got 9 as being the most optimal, it was still our experiment still correlated with our hypothesis, because our hypothesis was that the higher the pH, the more optimal, and the more acidic, the less optimal. And then the p-test obviously showed that a pH of 9 is also an optimum amount for trypsin activity. And now we are moving on to the discussion part. So this is our discussion. Pause here for a second so that make sure that you can get that. Our discussion. So basically, our group hypothesized that, again, if we change the pH of trypsin to a more acidic pH, then the trypsin will not be able to perform at its optimal. And our results were adequate with this hypothesis. And our results depicted in our table of 4 trials with varying pH ranges, that a pH from 4 to 9 is what we tested, and that out of those, the 8 to 9 tested at an average of 0.453 and 473, which is statistically significant and were the most optimal pH for trypsin enzyme function, because those pH levels are most similar to that of pH located in trypsin's natural environment, which is in animals, again, the small intestine. And while the pH of 4 had the worst level of enzyme activity at an average of 0.033 nm, which was consistent with our hypothesis that the more acidic the pH, it would more likely hinder the ability of trypsin to break down the azocasin. And then the negative control, which was tested without the presence of trypsin to break down the azocasin, also tested at a similar level of 0.027 nm, which exemplifies how little enzyme activity occurred at the acidic pH, especially at the pH of 4. And the groups that conducted this same experiment using acidity all had results that aligned with ours, that the lower the pH level, such as 4 or 5, had little trypsin activity, and that 8 and 9 had the most trypsin activity. pH was most suitable variable to test trypsin activity due to its level of influence in trypsin's natural environment. Also, testing temperature as the control variable at a temperature which mirrored trypsin's natural environment helped to test trypsin as close to its natural setting as possible. Possible variables that could have affected the experiment are incorrect pipette measurements, contaminating, affecting absorbance levels, and temperature fluctuations during the heating baths when groups would open the lids and take extended periods of time to place their samples in, causing the bath's temperature to drop. If the experiment were conducted again, groups could each use their own heating baths to help keep their desired temperatures for testing trypsin activity constant, instead of groups sharing heating baths, which could fluctuate temperature due to prolonged periods of time where the lid is open and the heat is escaping. Our final result for the experiment, along with the surrounding group's testing pH levels, expressed that the trypsin operates best at a pH between 8 and 9 and a temperature of about 37 degrees. And then lastly, just like trypsin, bacteria has ideal conditions for growth. And so that's why this experiment can relate to bacteria and what we will do with it in the future. That is all. Thank you so much. And this is the references that were used in the introduction. Thank you.